Okay, so chapter 7, it's about controlling microbial growth. So in the previous chapter, we already saw the way that some bacteria grows, and then we saw the growth curve, for instance, and now we're going to see how do you control that microbial growth. So in this image, you can see a scanning electron micrograph of a virus that causes gastroenteritis. It's very common to see it. It's norovirus. These typically contaminate water sources and is part of the outbreaks. It's actually a number one cause of gastroenteritis, virally related in the United States. So sanitation is very important when it comes to control microbial growth, and then part of it is checking these sources, these common sources that people uses for getting water, getting food, so you can control microbial population and then help to prevent having any outbreaks within the population. So we're going to see in this first part of the chapter some terms that are related to microbial control like sterilization, disinfection, antisepsis, degerming, sanitization, biocide, germicide, bacteriostasis, and asepsis. So whenever you hear the word sepsis, septic, that means that there is bacterial contamination. Septic tanks. It's a tank with microbial contamination, for instance. Asepsis is actually the opposite of sepsis. It's the absence of significant contamination. That is different from sterile. In a sterile environment, you don't have any contamination. And then part of asepsis is the aseptic technique that you use in the lab, but also when it comes to health, we have aseptic surgery that prevents microbial contamination from wounds. Now, sterilization is completely removal of microorganisms, all microbial life. And then we have different processes depending on what you're trying to achieve. For instance, for canned goods in the food industry, you use commercial sterilization in which you want to kill a major pathogen, which is Clostridium botulinum. This bacteria, it's a gram-positive bacteria, it's an anaerobe, it's part of the Clostridia species. Clostridia species are obligate anaerobes, are gram-positives. They are rod-shaped, and they have several members that actually cause several infections. Botulism, it's an infection in which you have flaccid paralysis of your muscles. So your muscles cannot contract because this bacteria produces a toxin that is called botulinum toxin that it prevents the actually neurotransmission between the muscle cells and the nerve cells. So that leads to flaccid paralysis. In the cosmetic industry, this type of toxin has been used for preventing someone who looks so wrinkly, Botox. So it uses these toxins, but when it comes to health, of course, it can affect our health. And now, then some canned goods, whenever they have endospores of an anaerobe, these anaerobes can grow inside these cans because just remember, when you can a good, you seal this can, and that leads to anaerobiosis. So that's basically the perfect environment for anaerobes to grow. And in that case, then As the anaerobes are growing in these cans, what they do is they produce gas, and this gas can be seen as a bulge. Not to the point that the can is going to explode uh, because of excess of accumulation of the gas. Eventually, the anaerobes will get destroyed, but by this absence of a good environment, because they will accumulate their own waste products. However, they will leave the toxins. And then when someone consumes a canned good that is filled with these anaerobes, of course the person can have a potential infection. And then disinfection. Disinfection is destroying harmful microorganisms on inanimate surfaces or environments. So if you apply a spray of disinfectant in a countertop, doorknob, seat, pencil, that will be disinfection. It has to be something inert, without life. And then antisepsis is actually destroying microorganisms from living tissue. Alcohol can be used for disinfection, but also as an antiseptic. Because you can rub alcohol in a countertop trying to destroy a virus, for instance, and then when you are going to have an intramuscular injection, that rubbing of the alcohol in your skin, that's part of the antisepsis. Degerming, it is the removal of microbes from a limited area. And it's mechanical removal. Sanitization is lowering the microbial counts on eating utensils to a safe level. This process is usual from restaurants, and restaurants typically, not the ones from fast food, of course, the ones where you eat in a plate and using utensils, they have like sinks with three openings. So one is for getting, well, they have, of course, they have like a waste disposal where you throw away the excess of food, and then you have one where you fill this sink part with soapy water, and it has to be at certain temperature. The concentration of the soap, it has to be also in a specific amount. And then next you have the rinse with water plenty, and it also has to be at certain temperature, that water. And then the next, they have basically the last rinse, but in this case it's going to be with the disinfectant. Now, if it's a good restaurant, they will control so much all of this so that you not only have the number of microorganisms on the eating utensils to a safe level, but sometimes they have so good sanitization that you don't have microorganisms on the eating utensils, especially when they put these dishes in the dishwasher. Dishwashers have a cycle at a specific temperature as well that can sanitize perfectly these utensils. And now the chemicals that we use to control microbial growth can be subclassified into two different types: the ones that kills the microorganisms and the ones that prevents the growth of microorganisms. So the ones that kill the microorganisms end in this ending site, and the general name can be biocide or germicide. So that's for killing microbes. Now, for preventing the growth of microbes, it is bacteriostasis, bacteriostatic agents. The preference for use one or the other will depend on the person. And then also the chemical nature of the agent. Not all the agents are bactericidal or virucidal, fungicidal. That's more specific for killing specific microorganisms. But some of them, by their chemical nature, they don't have this structure to destroy the microorganisms completely. And it's not ideal in so many cases, but that's possibly something that first you don't have control over, and second, might be the only thing available, and it's better than nothing. So, the usual definition of sterilization is the removal or destruction of all forms of microbial life. How could there be practical exceptions to this simple definition? Practical exception for removal of all forms of microbial life. So someone may try to possibly reach partial sterilization, trying to select for a microbe only. That's what. To be honest, I would say that there is no practical exception. I mean, that's not my question, of course. But, I mean, imagine if we were some instruments that are going to be used in a surgery, not only for a human, for an animal. Would you like to use contaminated instruments? No. And I guess I will say that maybe what they're trying to get is if there is some items that you don't want to sterilize. So what can that be? Maybe certain foods, right? Like if you use a lot of heat, you can destroy the nutrients completely, and you rather run the risk of eat whatever you have to eat. I mean, you trust that your acids in your stomach, your antibodies that your body produces, get rid of any microbes.
Like needles, like they can't like sterilize it, like they just throw them away and like get a new one for each person.
Yeah, in that case, yes. I mean, if there are certain things that doesn't necessarily need to be sterilized because they're single use. So why would you want to reuse a needle? There used to be a time, guys, believe it or not, early 70s when they used to, like nurses, they used to boil the needles and use them in people. Kind of crazy. For today, of course. So now we're going to describe the patterns of microbial death caused by treatments with antimicrobials. So we saw before in the growth curve how you have exponential death of microbes, just as you have exponential growth during the log phase of the microbial growth curve. Now, typically, when you have these microorganisms in a surface area that you're trying to control the growth, you start sterilizing, like let's say for 1 minute, and then depending on your method, but some methods they can kill only 90% of the population, and that's kind of theoretically a number or theoretical number, but you just want to make sure that if it's something that it should be sterile for sure, it will be sterile. So in this case, we have at minute 1 in this table showing you that out of 1 million bacteria or microorganisms that you start on time 0, you kill 90%, and then you end up with 100,000. Next minute, you kill that 90%, and then you end up with 10,000. And you keep on going until at minute 6, you have 1 survivor. So, something that it is important, if you remember for whenever we saw in the previous chapter, that graph and that table where it shows that out of 1 microbe, in the 5th generation, you end up with many. Well, what it tells you practically here is that you cannot stop the process at 6 minutes if it's something that kills 90% of the population every time, and then use that instrument or that device without thinking that you would have a consequence. Because this one, by binary fission, it will give you millions within minutes. So here we have the microbial death curve. We have this graph showing you the left y-axis, the number of microorganisms counted by the log 10, and in here, the arithmetic number of cells that you have, and then in the x-axis, you have the time per minute. And then the blue line represents this logarithmic scale, arithmetic scale, while this one represents the logarithmic scale, the one in the red line. So 1 log decrease, in this case, means 90% of the population decreasing. So again, if you take into account just these arithmetic numbers, you can think about that by 3 minutes you don't have any growth or any survival of bacteria after you treat this potential instrument or device for 3 minutes. But with this logarithmic scale, you can tell much better that you have a constant rate of death, at least 90%. So you don't have any drops or any increases. Now, the rate of microbial death will be depending on several factors. One of it will be how many numbers of microbes you start with. Second, the environment. Are these microbes protected by organic matter? Are they found in the right temperature? Do they form part of a biofilm? And how long do you expose these microbes to these disinfectants or these chemical agents for controlling microbial death? And then the characteristics of the microbes. Is this microbe more resistant than others? Is this microbe produces endospores? Is it a gram negative? Is it a gram positive? Is it a protozoan if it's a virus? I always give the illustration about perhaps someone being in their youth, adolescent years, and I'm pretty sure that that happens to many people. How many times you were in middle school and they started giving you the chores of doing your own laundry? And then you keep it for the entire week. And then perhaps during your sport practices, I mean, you had pant that it was soiled with soil and grass, and then you leave it there for a long time, and then you never pre-treat it. At the end of the week, when your parents found that you had all that pile of laundry in the basket, they called your attention and say, well, you better do your laundry, or you're not going to go out in the weekend. And then, of course, we are in that period of time where it's hard to be self-motivated, disciplined. And then instead of doing several loads of laundry, you do only one. How do you think the laundry is going to come? Clean? More than likely not. So you have a lot of microorganisms there. And then if you have that biofilm from having a lot of time there, these microbes, plus the organic matter that is in your garments, of course, you don't expect as efficacious results as if you were more careful. So here we have two type of examples. So same graph, but now we're using the logarithmic scale. So we have two population loads, one with high population load of microbes, another one with low population load. And if these were surgical instruments, well, if you start with this high population load, which has 6 log to 10 number of microbes versus 3, it would take you twice the time to reduce this to at least one survivor. So in this case, for the low population load, it takes you 3 minutes, and then in here it takes you 6 minutes. In the case of the laundry, not because someone does one load only for a bunch of clothing, and they put it to the maximum 120 minutes, that still is not going to come out clean. So, but here you have the idea. So the more microbes that you have, the longer that you have to spend or treat it with whatever method you're using. So check your understanding. How is it possible that a solution containing 1 million bacteria would take longer to sterilize than one containing 500,000 bacteria?
Because they each go down 90%, and 90% of 1 million is a lot more than 90% of 500,000.
Yes. So what is 90% of 500,000? 450,000? Yes. Okay, now we're going to see the effects of microbial growth agents on cellular structure. So we're going to see specific examples. And what do they do? So disinfectants or agents, sterilization agents, they can damage the plasma membrane, they can damage the proteins, or they can damage the nucleic acids. Damage to the plasma membrane would cause leakage of cellular contents because you create pores or big gaps in it. And that will interfere with cell growth because remember, when the plasma membrane it is damaged, doesn't interact well with the cell wall, and that leads to a cell death signal. Plus, let's say that death signal didn't happen. If you have a big gap in the plasma membrane, more content goes out than in. So you have dehydration of the bacteria or the microorganism. A window broken in your car. Turn on the AC all the way. You go to an area where it's super hot. That is not going to do any good. So similarly, you do with these microorganisms. You damage the plasma membrane, more content leaks out, it comes in. Damage to the proteins, enzymes specifically. If you damage the proteins, the enzymes, the metabolism of the microbes is gone. And damage to the nucleic acids. You can break down DNA, RNA, and then you will have consequences. So that's how you have these actions of these disinfectants. Now, does a chemical that is used for control microbial agents affect the plasma membranes in humans? How different are the plasma membrane of bacteria from ours? I mean, we don't have this LPS, right? Or the teichoic acids that gram-positive and gram-negative have. But the rest of the contents, do they have the bacteria proteins like channels? Do we have these channels as well? Yes. What about the phospholipids? So then, will a chemical agent that affects the plasma membrane microbes can affect our plasma membrane cells? Yes. Yes. Some disinfectants also are germicides in our skin, like quaternary ammonia, for instance. It's very irritant. You don't use it as much, but in a mild form or concentration, it can be used. So now let's look at physical methods for controlling microbial growth. So we're going to see process of moist heat, dry heat. We're going to see filtration, low temperatures, high pressure, desiccation, and osmotic pressure. And then we're going to see radiation. So all of these are physical methods. It's going to be very familiar to you because you already saw part of this in the previous chapter. Mode of action of heat. It denatures enzymes. Denaturing proteins, which includes enzymes, of course, it will be unfolding the proteins that basically destroys the three-dimensional shape that proteins have, and then the enzymes won't work. When you use heat, you can control how much heat you use by taking into account thermal dead point and thermal dead time. Thermal dead point, it is the lowest temperature that is necessary to kill all cells in a liquid culture when you use that heat for 10 minutes. So it's the minimum temperature to kill all the microbes within 10 minutes. Thermal dead time, it is the minimal time for all bacteria to be in a liquid culture to be killed at a particular temperature. So TDP, it is related to the minimal temperature, and then thermal dead time refers to the minimal time. Now we have decimal reduction time, which it is the number of minutes that takes to kill 90% of a specific population at a given temperature. So what you saw before in that graph is DRT, decimal reduction time. And this is based on statistics, and it is based on the fact that, again, you have to make sure that you go beyond having one cell. You can say, well, I use the TDP, thermal dead point, and then I believe that all the cells were killed within 10 minutes. Does it hurt so much to sterilize something that can be killed in 10 minutes 100%? Does it hurt to run it for one more minute? No, it doesn't. In this case, you're trying to protect something. Maybe it's your experiment that you're doing. You're autoclaving some beakers, or some pipetters that are autoclavable. So you want to make sure that you go beyond what it could be 100%. That's why you have this DRT, the 90%. So that's the general information about heat. Now, for moist heat sterilization, the first type of heat that we can use, so, or well, that we are considering. So moist heat, it denatures the proteins just as heat. And another way to say denaturing is coagulation. And we have boiling. We have free-flowing steam. And free-flowing steam, typically it is done in the autoclave. And boiling, of course, it is boiling water, for instance. So for the autoclave, we have specific parameters that you have to follow. No matter that you might have a thermal dead point of 10 minutes, we have, for instance, in these autoclave machines, we have a minimal temperature of 121 degrees Celsius. And you have steam produced by this boiling of the water, and then you will close the chamber, and the water, as it's forming vapor, is going to accumulate this steam, this vapor, and then you will increase the pressure. The minimum pressure is going to be 15 pounds per square inch, and then you will use a minimum time of 15 minutes. Why is this? Well, because with the exception of prions, which are proteins, you kill all organisms and endospores at this temperature, pressure, and time. Something for sure is that the steam must get in contact with this surface of these items. And then this process is highly regarded in healthcare and lab facilities as well, because it's very effective. Here is the internal view of an autoclave. So this is a chamber made out of walls of steel. Here you have your door that has a gasket that hermetically seals this opening. And then you have some ports through which you will introduce steam into the chamber, and that steam is going to go through a pressure gauge to tell you how much pressure you have. You have a safety valve just in case that you need to stop the process during this time where you're running the autoclave. And then you have a port where you can exhaust this steam after your sterilization process is done, and this steam goes into a wasteline. Now, some chambers have already water supply on their own, but at least in the one that we have here, we have to put the water inside the chamber. And it has to be a specific amount of water. In this case, for us, it's 4 liters. If you have less water than that, the autoclave won't run. And then some of them, they also have automatic waistlines where the water goes through the drain pipes. And in some other ones, you have to drain it manually. And then when the right pressure is found within this chamber, if you have, for instance, in this case, liquid media inside this chamber, you have to have a cotton plug covering this opening of the flask, or you may have that plus like a little lid that you have made with aluminum foil. And this is to help for partially the steam go through this aluminum foil and then go inside into this liquid media so that you can destroy any microorganisms that are there. And then once the steam, once the time has passed by and the steam will be released from the chamber, you have gradually reduction of the pressure that is accumulated inside. And then you cannot open the chamber, the autoclave, unless that pressure is 0. First, the machine is not going to let you open the door if it's not 0. And second, you run the risk of burning yourself because that pressure is going to blow the steam against your face and your body. Now, when it comes to temperature, you also have to wait for the temperature to drop. It's not only the pressure, but also the temperature. So there are specific parameters that it will be important to follow according to where you will be working with these machines. So, as usual, you understand that large containers will take longer process of sterilization. And then depending on what you are sterilizing, if you're sterilizing waste, you will need perhaps to increase the time from 15 minutes to more. And then you have some indicators of a sterilization process, like test strips, that either can tell you the name, telling, well, yes, a sterilization has taken place, or maybe you will use this tape that has these white lines in an angle that once you sterilize your materials, it will change from white color to dark color. So this is what it changes in terms of volume of liquid that you're using, and of course, the flask that you're using. So in this case, we have several examples in this table, test tubes that are 18 millimeters times 150 millimeters. They can hold up to 10 milliliters of liquid media. So in this case, the sterilization time is 15 minutes. But when it comes to sterilizing like 1.5 liters, in an Erlenmeyer flask that is 2 liters, sterilization increases to 30 minutes. And then of course, if you have large volumes, like in this case almost 7 liters, it takes you 70 minutes. So here is an example of a strip, an indicator for sterilization. So they introduced these 2 strips into the autoclave. This one was protected completely by this aluminum foil. They didn't, I bet they put it in a way that no steam went through. And then in this case, they put it outside of a foil, unprotected. So meaning like the steam reached into here, and then this is previous to sterilization. So when you put these strips, they say not sterilized. And then if you didn't have the contact of the steam, it will continue like this. But something that perhaps was sterilized and you use this indicator, in this case, you have sterilized. So this yellow color changes to blue or dark color, and then that omit the thing of this word. Any any questions so far? Okay, so that was part of moist heat sterilization. Now, another type of heat sterilization is pasteurization. So in this process, you will have the reduction of microorganisms that can spoil the food, and also pathogens from milk and juices. We have 2 types of pasteurization. We have high temperature, short time, which brings items to high temperature really quick, 72 degrees Celsius for 15 seconds. And pathogens will be killed, but organisms that are what we call thermoduric survive. These can spoil refrigerated milk after a period of time, but they don't cause disease. So high temperature, short time. After this short time, you bring the juices or milk to a low temperature, also very quick. Now, the next type of pasteurization is UHT. It's ultra high temperature. In this case, it's almost twice the temperature from HTST. So it will be, instead of 72, 140 degrees Celsius. And it's 1/3 of the time from the previous one. The other one was 15 seconds. In this case, it's 4 seconds. And similarly, it's followed by rapid cooling. So this will also sterilize milk, juice, and creamers. And an advantage of this is that because you use very high temperature, you can even kill the thermoduric bacteria. So then you can leave these items at room temperature after they've been subject to this process in a carton container. Of course, once you open it, the sterilization process is gone, so you have to put them in the fridge if you don't use the entire product. So this is helpful for going on a field trip. You want to have a juice that won't get spoiled, milk, you can bring it, and then you use it, and then you still have your nutrients there. Dry heat sterilization. So dry heat sterilization, it is used for killing by oxidation. Examples will be flaming. like what we do with these loops in the uh, in the lab. Oftentimes, when some sheets from the beds are contaminated by pests, uh, usually people they have to burn their sheets. So that's incineration, flaming. And then we have hot air sterilization, in which you have an oven that can bring your items up to 170 degrees Celsius, and then you will sterilize your items typically for 2 hours. These are sterilizers that are used in some healthcare facilities, for instance, dentists. Sometimes they have these ovens. They sterilize their instruments in these ovens. Having an autoclave, it's a big responsibility. Not everyone can have an autoclave. I mean, these Instant Pot guys are autoclaves. And well, at least thankfully these ones, they can be controlled for the time, and then you can go away and then nothing happens. But what if you're at home, you have like these other types of pressure cookers, and then you leave home, you leave it there after you're gone on in the stove. What will happen if you come back home after a while? More than what you're supposed to have, leave it there. Mm-hmm. Will explode. So we have mini autoclaves at home. So big autoclaves are definitely something that it needs to be taken care either in a university or a hospital, somewhere where people is trained to use them. Now for filtration, this is another physical method for control microbial growth. It is passing a liquid substance through a screen-like material. It is used to sterilize or to control the microbial growth of heat-sensitive materials. Like for instance, HEPA filters. They are used to sterilize the air. It has pores that are useful for trap microorganisms or particles greater than 0.3 microns in diameter. And then for membrane filters, we have some that they remove microorganisms that are greater than 0.22 micrometers, and some of them, there can be super small, like the ones that can filter out viruses and large proteins that can measure 0.05 micrometers in diameter. Water is usually filtrated. Why? Well, because you can have these viruses contaminating the water or protozoans. Not everything, especially potable water, it is killed by chlorine. Some microorganisms are chlorine resistant. If you go to a pool, you can tell, well, I mean, water is sterile because they put a lot of chlorine. You can even smell it. But cryptosporidium parvum, it's a protozoan, the cysts. They're too tiny that if they don't filtrate it, they can be in the water. I mean, who cares? Well, an immunosuppressed person cares about it. Why? Because these produce severe diarrhea that the person can die of dehydration. If you have been in a swimming pool, I don't think, without using perhaps something that it's super protective in your mouth or your nose, I don't think that no one here hasn't never drank water out of a pool. And this is not to gross you out, but people do crazy things in pools.
Yeah.
So imagine if someone has, if it's an immunocompetent person, it has Cryptosporidium parvum in their body. They have an accident, and then someone ingests this. And again, this protozoan is chlorine resistant. So then, if it's an immunocompromised person, that person can die out of that infection. So that's why they filter the water in the pools besides putting chlorine. And I'm hoping that. So here we have an example of filter sterilization in the lab. So we have dual chambers in here. These chambers, the one in the bottom is the one that is going to catch anything that is microorganism free. They have a port that it can be connected to a vacuum line to help for extraction of this fluid from the top chamber. So then you pour your media here, and then the media goes through this filter, and then anything in here is supposed to be sterile. So when do you use this type of sterilization? When some of your ingredients are heat labeled, so they can be destroyed by this by the heat. Examples: vitamins, amino acids, maybe even antibiotics. So you can have all your ingredients here pass through the filter and you don't destroy them. There are companies that they sell these pre-made media, but basically that's what they do. They filter it in this manner, they sell it to you. But let's say that out of one flask that you have 500 milliliters, you use 250 to grow a cell line. So in this case, let's say that you want to use the other 250, because when you work with cell lines, the material that you use for keeping these cell lines going is expensive, and the media as well, of course. So what if you want to use these 250 milliliters, you use 250, you don't want to throw away, like, let's say $800 that can cost. So in this case, you may have a filter there, and then you can pass your media through the filter. And then you have to check, of course, when you're looking at your cell lines, that you don't see any contaminants, that the cells are happy. Another way to control growth of microorganisms is by using low temperature storage. That's in our daily lives, not necessarily in the lab. So refrigerators are used to prevent the spoilage of food. It's okay. I think it's fine. Like when people, they buy their groceries, their vegetables, their fruits, they wash them and then they put them in the fridge. But sometimes it's kind of impractical in a way. I guess we put them there, and then until you consume, you will wash them. And of course, if it is ones that are pre-washed, you don't do that. But in essence, let's say that your items that you buy at the grocery store are contaminated. I never seen, and I don't think it will exist, of course, that you're picking up your groceries, an orange falls, an apple falls, and then you just pick it up and put it in your plastic bag or your container. Now you put it back in the pile and you grab something else, but you don't know how many times has fallen before. And if I own a grocery store, I guess I will have like a tiny container there on the side and say, well, if this one has fallen, put it in there. No one does that. So, so I don't trust, of course, knowing myself. But then you put your items in the refrigerator, hoping, of course, that your refrigerator is working well and then you don't have moldy things going on. And then for the lab, of course, you keep your stocks in the refrigerator. And then for deep freezing, I explained to you this method. Basically, you have a -70, -55 freezer that is frost-free, and then you don't have the cycle frosting time, and that preserves your microbes for long periods of time when you prepare them correctly. And then lyophilization. That's the ultimate way of preserving your microorganisms. Not everything can be lyophilized, but the ones that they can be lyophilized, you use freeze drying first, and then you put them in an ampule where you can preserve them at room temperature forever until you hydrate these microorganisms again, this powder. And now you can use also high pressure that denatures proteins and alters carbohydrate structure. Some of these methods are used in the food industry when you cannot use heat treatment. For instance, sometimes they can do that with avocados to keep these. If you make a guacamole, to keep this free of microbes for long periods of time without spoiling. And then desiccation. It is used to remove water from the microorganism. And remember, without water, in general, any cell, the metabolism drops. So then desiccation is one method. How we desiccate? By having these items put in a high concentration of salt or sugars, so you create a hypertonic environment. And since you have more solids outside the cells than in, the water goes out, and then the cells dehydrate. and thinking about that, halophiles, they like this high concentration of salt. So last physical method, ionizing radiation. So we have several types. The use of X-rays, gamma rays, which both of them, they have high energy. They can penetrate to the items, break down macromolecules like DNA, creating mutations in the microorganisms. And then we have electron beams. So when you use this, something as well that you can create, it is hydroxyl radical. So you break down water particles, and then hydroxyl will pick up an extra electron, and that can jump between these different macromolecules, also creating mutations and destroying proteins. So about gamma rays, these ones are very powerful, penetrate deeply, but it requires several hours for sterilization. And then for these high energy electron beams, they have less penetration, but it is fast because these electrons really easily move between the molecules. So these as well are used, these methods, in the food industry, like to preserve spices, certain meats and vegetables. Again, this can be used for avocados, for instance, and for sterilizing pharmaceuticals, dental and medical supplies, like our the needles, the syringes, and also for mail. we can stop here. So any questions?